This experiment aims to understand the batch kinetics of cell cultures. Two nutrient media, LB and M9, and two reactors, benchtop shaker and bioreactor, are used. Kinetic parameters are determined and compared for each operating condition. Bioreactors and cell cultures are used in a variety of industrial processes. Understanding the kinetics of these systems aids in optimization of cell growth. E. coli is a suitable bacteria for investigation because of its consistent growth behavior and close adherence to proposed models. There are four phases of bacterial growth in a batch process. The first is known as the lag phase, which occurs when microbes are still adapting to their new environment. Population growth is negligible during this phase. Phase 2 is known as the log or exponential phase, which describes the period of optimal population growth. During this phase, microbes are consuming available substrate and growing at their maximum rate. Phase 3, or the stationary phase, occurs when the population reaches its upper limit of growth, also known as the carrying capacity. The microbes will run out of nutrients or space if the population grows beyond the carrying capacity. The final phase, called the death phase, is a population decline that occurs when cells begin to die due to a lack of substrate to consume. The respective rates of change of biomass and substrate concentration can be described by equations 1 and 2. The growth rate parameter, mu, can be described as a function of substrate concentration, as shown by equation 3. This is an example of the Monod model, not accounting for cell death. Determination of the kinetic parameters Ks and mu max requires a different approach depending upon the experiment. Due to the fact that Lb is rich in nutrients, it can be assumed that substrate concentration remains constant relative to changing biomass. For this reason, Ks cannot be determined for Lb media. Umax can be determined from the semi-log plot of substrate concentration versus time. For M9 media, there are two ways to determine the kinetic parameters. The first method is similar to what was used for Lb media. Ks is found from substitution into other model equations. The second method is to plot 1 over growth rate versus 1 over substrate concentration. Both methods can be applied and the results compared. E. coli is a highly toxic biologically active material. It is important to have a sterile environment in order to prevent contamination and contact. An autoclave was used to sterilize equipment and glassware before use. Biological materials should be handled under a fume hood when possible. In order to keep the fume hood and other working surfaces sterile, spray down gloved hands and glassware with ethanol before use. When finished with the work area, spray down with bleach, ethanol, and water. Dispose of cell cultures in an ethanol waste container. When the reaction is completed, add bleach to the reactor vessel and sterilize for 30 minutes before disposal. After disposing of biologically active material, place used glassware and containers in a wash basin of bleach solution. In order to allow the bacteria to adapt to the media, it must be inoculated about 15 hours before running the reactor. Both reactors follow the same inoculation procedure. Measure 60 milliliters of room temperature media into a graduated cylinder and transfer to a 125 milliliter Erlenmeyer flask. Warm a sample of bacteria until it becomes liquid and pipette 1 milliliter into the flask. Dispose of bacteria containers and pipette tips in the sharps container. Cover the flask and let stand overnight. Measure 95 milliliters of media into a graduated cylinder and pour into a 250 milliliter flask. Pipette 5 milliliters of inoculated sample into the flask of media. Set the benchtop shaker to operate at 37 Celsius. The cells die at 40 Celsius, so temperature should be monitored closely. Set the shake rate to 175 RPM. Wipe down the bottom of the flask and press firmly onto the shaker surface to ensure the flask stays in place. In order to determine biomass concentration of the sample, the optical density at 600 nanometers is measured. Every 30 minutes, remove the flask from the shaker. Move it to the hood and pipette 1 milliliter of sample into a cuvette. Zero a spectrophotometer with a cuvette of pure media. Measure the absorbance of the sample, keeping in mind the direction of the light beam, and record the value. Return the flask to the shaker as quickly as possible to maintain proper operating conditions. Repeat until consecutive data points are identical or until the reaction has run for 8 hours. Place the reactor on the control system. Attach the inlet and outlet water tubing for heating the reactor.
Good. Oh, there we go.
Attach the agitation motor to the reactor assembly. Run the temperature probe into the reactor. It must reach the bottom of the reactor to measure the sample temperature and not ambient temperature. Turn on the water source above a bucket for catching leaks. Select the cell culture option on the reactor controls. Program the temperature and agitation rate to the desired values. Use conditions consistent with the benchtop shaker. Pour 950 milliliters of media into the reactor. Once the media has reached the set temperature, pour 50 milliliters of the inoculated sample into a graduated cylinder. Pour the sample into the reactor using the same funnel. At the start of the reaction and every 30 minutes after, use a syringe to withdraw sample from the reactor. Transfer the sample to a cuvette and measure the absorbance using a spectrophotometer previously zeroed with media as shown in the benchtop procedure. Repeat until consecutive data points are identical or until the reaction has run for 8 hours. When comparing kinetic parameters with LB media, it was found that the maximum growth rate was greater for the bioreactor. This is expected because of drops in temperature and agitation when sampling the shaker flasks, which may inhibit the maximum growth rate. Additionally, the bioreactor has more available oxygen than the flasks, which contributes to greater cell maximum growth rate. It was found that the maximum growth rate was greater for LB media than for M9 media. This is due to the fact that LB media is richer in nutrients. As a result, the carrying capacity of the cells will be greater for the LB media. Method 1 for calculating the M9 kinetic parameters yielded reasonable values. The parameters from method 2 were negative and therefore not meaningful. This could be the result of insufficient number of data points or inaccurate data. Experimental M9 data of biomass and substrate concentration was plotted versus time. This was compared to a numerical MATLAB model of monode kinetics using parameters from method 1. It was found that the MATLAB model represented only the exponential phase, while the experimental data showed a stationary phase. The bioreactor was shown to have higher growth rates than the benchtop shaker due to more consistent operating conditions and greater exposure to growth-inducing oxygen. LB media was found to cause higher growth rates than M9 media due to a higher availability of nutrients for cell growth. The MATLAB model did not accurately represent the change in biomass and substrate concentration over time for M9 media. This is because the model equations only accounted for the exponential phase.